This is the journey of organic molecules by Sadie and Gwendolyn. Meet 9-year-old Sammy. Sammy has a few questions about food. So, what does food actually contain? How does his body use it? Where does his food travel through his body? And why are some foods considered healthier than others?
So Sammy's favorite food is peanut butter, so he was wondering what makes it so good and what do the ingredients on the nutrition label really mean. So peanut butter is mainly made from plants. It has 190 calories. Calories are a way to measure the energy content in food and drinks. There are 9 calories per gram for lipids and 4 calories per gram for carbohydrates and 4 calories per gram for proteins. And peanut butter has 16 grams of fats, 8 grams of carbohydrates, and 7 grams of proteins.
So, when food is tested for carbohydrates, lipids, proteins, and simple sugars, they use these tests. So, for proteins, a positive test shows a color change from yellow to blue-violet. Simple sugars, a positive test shows a color change to red, orange, yellow, or green. Carbohydrates, a positive test shows color change from yellow-brown to blue-black. And for lipids, there's 2 tests. One of them is the Sudan-3 solution, which turns red in the presence of lipids, and the paper test, which shows when you smear the food onto a piece of paper and hold it up to the light, a grease stain shines through.
So the first test is the iodine solution, which is for carbohydrates. And so it's supposed to turn blue-black, and ours turned a very camouflage green, so we concluded that there was a small percentage of carbohydrates.
Okay, so the next test was the Sudan stain, and this test for lipids. So for the Sudan-3 solution, it turns red in the presence of lipids, and when we tested peanut butter, it turned a pink, so we concluded that there was lipids, but not a strong percentage. Another test that we did was the paper test, and when we smeared peanut butter onto the paper and we held it up to light, a grease stain did shine through, so we knew that lipids were in peanut butter.
The next test was the Biarritz reagent. It was for proteins, so it was supposed to change from yellow to blue-violet, and ours turned a dark blue.
One interesting observation that we made was when we put the solution into our peanut butter, when it first made contact, it was a very, very dark blue, but then once it started to diffuse, it just became a darker blue. Our final test was the Benedict's reagent, which tests for simple sugars. Okay, so this right here is our representation of a glucose molecule, which is a simple sugar.
So the simple sugars test was the Benedict solution, and it was supposed to, in the presence of a simple sugar, change to red, orange, yellow, or green, and our solution turned into a brown-orange color that was kind of dark, so we concluded that it did contain a small percentage of simple sugars.
Okay, so ingestion. Ingestion is the first step in the journey of peanut butter. So ingestion is when the food, or in this case peanut butter, is just put in the mouth. There is no breaking down of any molecules at this point.
Next step after ingestion is digestion, where the food is passing through the digestive tract, which is the stomach and the small intestine, and it is being broken down into its subunits, as you can see by this picture.
Okay, so your mouth is the first stop for digestion. This is where mechanical digestion takes place because your teeth are chewing your food into smaller pieces to increase surface area for absorption.
Other than mechanical digestion, chemical digestion of starch also begins in the mouth because of an enzyme called amylase.
Okay, so enzymes make it easier to break down and build up molecules. However, in this case, amylase is beginning to break down starch into the glucose subunits.
So these carbohydrates are being broken down into the glucose subunits, or the simple sugars, which are represented by these hexagons. And later, these glucose subunits are going to reassemble to create glycogen.
Glycogen is a long-chain molecule that will be used for storage in animals. In humans, glycogen is mostly stored in our liver and muscle cells.
The next step in the digestive system is the esophagus, which is the pipe leading from the mouth all the way to the stomach. And so these are representations of the molecules found in peanut butter, like lipids, proteins, and carbohydrates, going down the esophagus.
Okay, so in the esophagus, there are 2 layers of smooth muscles that contract to move the food down, also called a bolus. And there also is mucus that lines the muscles, so it reduces friction for when the food goes down the esophagus.
After the food exits the esophagus, it goes into the stomach, which is the next step of the digestive system. So there are three things in the stomach that help digest proteins. So there's mucus that's protecting the lining of the stomach and keeping everything in. Then there's pepsin, which is protease, which is an enzyme to break down and chemically digest proteins. And there's hydrochloric acid, or HCl, which is providing the acidic environment so that the pepsin will digest the proteins.
Okay, so pepsin is the main role in the digestion of the stomach for proteins because mucus and HCl just help pepsin digest the proteins. Okay, so this is the breakdown of proteins. So this right here represents proteins. Each of these different shapes represents different amino acids, like right here. So there are 22 different types of amino acids. 13 of them your body makes, and 9 of them are called essential amino acids, which can be found in protein-rich vegetables and animal sources.
After the stomach, the next step in the digestive system is the small intestine, which feels velvety because of the villi, which are finger-like extensions in the lining, and the villi are lined with microvilli, and these both help increase surface area for the absorption of nutrients, vitamins, and minerals. In the small intestine, there is a continued breakdown of carbohydrates into glucose subunits and proteins into amino acids.
Okay, so the breakdown of lipids. The breakdown of lipids happens in the small intestine, and they can be broken down into glycerol and fatty acids. And when they reassemble, they can become glycerol, fatty acids, and additionally to that, they can also become fats as a whole. So lipids are broken down into small pieces by bile, and lipase enzymes are the ones that break down lipids for chemical digestion.
And if you probably noticed, all the enzymes that are breaking down organic compounds end in ase, like protease, amylase, and lipase. And usually, with the exception of amylase, the first part of the word has to do with the molecule they are working with, like lipase, lipids, and protease, proteins. Another way to break down organic or chemical compounds, other than using protease, amylase, and lipase or other digestive enzymes, is hydrolysis. And hydrolysis is when an addition of water is used to break down compounds.
Okay, so as we said in the small intestine, there is villi and microvilli used for absorption. And when they are absorbed, these nutrients and molecules will go into the bloodstreams as we have here, and these are carried. These molecules and nutrients are carried throughout the body to cells where they are needed. This is called circulation. So, like as you can see, we have like glucose and different amino acids going to different places in the body to where they are needed in the cells.
So after all of the subunits are circulated into the typical cells, they are going to be reassembled into bigger and larger molecules. So carbohydrates are formed from some glucose in typical cells, and then fatty acids and glycerols can create new lipids, and different amino acids can create new proteins.
Okay, so in addition to growth, the reassembled molecules are also used for storage. So you can see plants and animals have different uses for them. Plants can use sugar molecules and reassemble them into cellulose for their cell wall structures. Plants also store carbohydrates by making glucose into starch for energy. Okay, so animals use proteins for structure, and as we said earlier, they also use glycogen for structure in their liver and muscle cells.
And so this is just an example of how cellulose is used to create the structure of a cell wall in a plant cell.
So after circulation, and all the molecules have gone to the typical cells, these are some of the roles of proteins. So proteins are made up of carbon, hydrogen, oxygen, and nitrogen. And the really interesting thing is proteins are the only organic molecule that have nitrogen. And so these elements help proteins have these uses, such as they control reactive reactions, they help form bones and muscles in the body, they transport substances in and out of the cell, and they also help fight disease.
So once lipids are in typical cells, they also have many uses. So lipids are made up of carbon, hydrogen, but very little oxygen, which is much less than carbohydrates and proteins contain. So lipids are used as part of biological membranes. They can also be used to store energy and also as chemical messengers.
Okay, so like lipids and proteins, carbohydrates also have a role in the body. Carbohydrates are made up of carbon, oxygen, and hydrogen. The carbohydrate subunits are used for a few things. For example, they are the main source of energy for living things, and plants and a few animals use them for strength and rigidity, like when we were talking about how they use cellulose for the plant cell wall structure. Cellulose is a complex carbohydrate.
So as we said, carbohydrates are used as the main source of energy in living things, but their subunits can also be used for energy. So in the process of cellular respiration, simple sugars or glucose molecules or glucose are used for energy. So the energy from the glucose or sugar is used to create ATP. So carbohydrates and their subunits, glucose, have many functions. So another of glucose is when they bond, they are extracting water. So this is when all the elements really come into play. When they are bonding, they are extracting the oxygen and hydrogen, which is creating water.
Okay, so the chemical formula for glucose is C6H12O6. So one of the molecules is losing a hydrogen, and the other one is losing an OH group, as you can see down here. So these two bond together to form H2O, or otherwise known as water, which is why it's called dehydration synthesis.
So to answer Sammy's questions, the first one is, what does food actually contain? And well, food contains carbohydrates, lipids, and proteins, but also calories, vitamins, and minerals, but those three do not fall into the three main categories.
So how does his body use it and why? His body uses it for energy, storage, growth, and structure. And why? So for example, proteins are organic compounds that can fold into many different complex shapes, so they have a wide range of variety for the functions in the body.
This third question is, where does my food travel? And your food travels from where it begins in the mouth, through the esophagus, through the stomach, small intestine, large intestine, into typical cells throughout the body, as well as coming out as waste.
Okay, so the final question is, why are some foods healthier than others? Well, your body needs different types of carbs, proteins, lipids, vitamins, and minerals. And all different types of food contain different amounts. So it's important that your body gets a wide variety so your body can function properly.